I think we can get rolling here. Welcome everybody to Rakovina Therapeutics and Variational AI's joint update, and with me today is Mads Dalgaard, who's the President and Chief Scientific Officer of Rakovina Therapeutics, and Handel Kim, who is the co-founder and CEO of Variational AI. And my name is Jeffrey Baccia, and I'm the Executive Chairman of Rakovina. Today's going to be interactive. We're going to talk a little bit about really how this all came together, what we've been doing together since we formed this collaboration over the last year or so. Some very exciting data that we are thrilled to have presented at the Society for Neuro-Oncology meeting that has come out of this and really is potentially very impactful for patients. We want this to be interactive, so we're going to reserve the last 20 minutes or so of the conversation for Q&A. You can feel free anywhere along the way to type your questions into the chat, and we'll go through them once we get to that point. But once we get into the Q&A, if you do think of something, just use your raise your hand function in the Zoom functionality, and we'll do our best to get to everybody. When you're asking a question, if you're comfortable turning your camera on, that would be fantastic. That way we can sort of see who we're talking to. It's not mandatory, but that would be appreciated. So today's really about two young and exciting Canadian companies combining forces in terms of kind of deep biology meets generative AI. So before we get into all of that, just wanted to set the table. So, Handel, for those who aren't familiar with Variational AI, why don't you give a snapshot, and then we'll go from there.
Sure, thanks, Jeff. Mads, it's great to be here, and thank you everyone who's on the line for joining. Variational AI, we were founded in 2019. We're based in Vancouver. We are 19 FTE, 13 PhDs. And the core team, the founding team of Variational AI came from a company called D-Wave Quantum, where we were the generative and probabilistic AI group. And then in 2019, we started Variational AI to bring generative AI into the industry of drug discovery, specifically in small molecules. Since then, we've made rapid progress in terms of taking our model that we call Enki and turning it into a platform that we use in order to generate novel leads. And for people who are maybe a little less familiar with the process of drug discovery versus drug development, and drug development is once you're in the clinic in your phase 1, phase 2, phase 3. Discovery happens before the preclinical side, the early stages of hit ID, hit to lead, and lead optimization. That's really where we live. And what Enki, our foundation model, is able to do is be able to skip hit ID and hit to lead and generate directly these lead structures, which we then do lead optimization for, thereby reducing the amount of time and the amount of cost dramatically, but more importantly, coming up with these novel molecules that are very, very hard to come up with. And that's what we do exceedingly well. We've done it for Rakovina. We're proud to be a partner there. And we've been able to generate these results in effectively one sort of cycle, which speaks to the power of the model and also the ability of Rakovina to choose good targets. You can think of us like snipers. You tell us what targets to hit, we'll hit them, and which targets not to hit. But we don't necessarily have the biology, and that's why we partner with world-class organizations like Rakovina. Otherwise, we're going to shoot bad targets. So we're only as good as the targets that you select. But we've done this for Rakovina. We've done it for a number of biotechs across North America, as well as a recent announcement with Merck for a $349 million biobucks deal that we announced in September. So we do this for pharma, and we also do this for biotech.
So Mads, I know a lot of people on the call know Rakovina fairly well, but why don't you give a quick overview and how to sort of that dovetails into to how we're working with Variational.
Yeah, absolutely. Hello, everyone. It's great to be here. Rakovina Therapeutics is a drug development company, and Rakovina focuses on DNA damage response pathways as targets for their drug development. It's also a young company, fairly young company. We are about 5 years in. And I just want to remind you that the focus on DNA damage response pathways is something that is extremely attractive in cancer research. The DNA damage response pathway consists of about 20 to 30 core proteins with very specific jobs in repairing DNA. And when cells encounter damage to DNA, then all these response proteins, they help the cell make a decision on whether to repair the damage or commit suicide by activating a cell death program. So the reason why DNA damage and repair proteins are interesting in a drug development context is that cancer cells, they often have mutations and deficiencies in their repair systems. And that creates a vulnerability for the cancer cells that we can exploit with drug development and targeting these pathways. So this is what Rakovina is doing. Rakovina is exploiting ways, new ways to cleverly target these DNA response pathways so we can preferentially harm cancer cells.
Good. Thanks, Matt. So, let's talk a little bit about how this all came together. We've got two teams, one that's deep in the wet lab, the other that's immersed in the cloud. I recall handle a meeting with you and your team at Bio a couple of years ago and starting some conversations about what ifs. But in reality, these types of collaborations are built on relationships, and we're all part of the Vancouver, British Columbia biotech ecosystem. So kind of all knew each other anyway. So why don't just sort of give the backstory about how this began to come together, and then we can talk about the successes that have happened.
Yeah, absolutely. I mean, we knew Mads from the Vancouver Prostate Center, and we collaborated with various folks there at VPC. We didn't know he was involved in Rakovina when we met you, Jeff. So, when we did meet, I believe it was in San Diego a couple of years ago at Bio. It was, when we met and you told us what you guys were working on, ATR and DDR, the DNA damage, response mechanism of action was something that we were quite familiar with. And we said, yeah, this looks great. And, bonus, you're in our backyard. So it cuts down on a lot of travel. So we were absolutely thrilled to be able to work with, a world-class organization, especially, the deep biology expertise that Mads and his team have at VPC. And so, again, we're snipers, and you just tell us what to hit. The better you can explain what it is that we need to hit, the better the outcome is going to be. And so, I think it's very much a marriage of two complementary strengths in biology and target. And the ability to create these novel, chemical entities that eventually, Rakovina will take forward and into the clinic, and then hopefully market post approval. So we're absolutely happy to be able to work with a great organization like what you've assembled over at Rakovina.
So, and Mads, I mean, we've been working together for well over a decade on various projects, and sort of having the opportunity to do something a little bit different with Variational, with an AI platform focused on kinases, which are sort of front and center in DNA damage repair. And talk a little bit about so the light bulb going off and wow, this is going to be amazing when we started talking about it.
Yeah, I mean, I think the synergy really happened when, we got interested in the kinase target called ATR a while back, and and it was obvious, I think, for us that ATR was a terrific target, And what we really wanted to do in Rakovina was to target not only ATR, but also target another kinase at the same time, called mTOR. And on top of that, we also wanted the drugs developed to be able to penetrate into the brain. And that is really a really tall order, because brain penetrance is a really big obstacle and problem in drug development. Our central nervous system and brain, they have this membrane around it. It's basically an encapsulation, a membrane encapsulation that protects our central nervous system and brain from all kinds of toxins and immune cells and bacteria and infections and all that. And that is, of course, all good, but it also creates a problem for drug development because most of the medicine that we have cannot cross into the central nervous system and in the brain. So we thought that AI and the absolutely fantastic platform that Variational AI and Handle has built here was really a fantastic way to approach this problem and combine all these features into a drug development approach. So that's really what brought us together, I think, for this project here. That we met over the problem. We wanted really to develop drugs with very certain specific characteristics, and Variational AI, they had the means to do it with the Enki platform. So yeah, so I think that's really the story on how we met.
I remember those early meetings when we were all sitting together, sort of around the table at Handel's offices and the computer lab, if you will, not the wet lab like Rakovina has. And besides the fantastic food truck that was parked outside that we were enjoying that day, I remember sitting around and looking at the list of kinases that were sort of front and center in DNA damage repair, and the input across the two teams in really narrowing down what we wanted to focus on. It was a really robust conversation, and the expertise on both sides of the table. And there is deep understanding of chemistry and biology on both sides. And I think just recalling that day and the roundtable discussion of, okay, the world's kind of our oyster here. We've got a list of things that we could do. What should it be, and how are we going to tackle it? So if you sort of rewind and talk, walk us a little bit through that day and then kind of what happened since then, because from that day, which was in the fall of 2024 into early 2025, there was a tremendous push that led to a big announcement in January last year, and we talked about how we got there and what we've done since.
Yeah, I mean, from our perspective, what you were looking for in the ATR, program was perfect in the sense that we specialize in ensuring that there's selectivity, and we specialize in ensuring that all the parameters that need to be met within a small molecule therapeutic can be optimized, simultaneously. And that's where we differ from traditional drug discovery, where you may have a target, let's say, for example, ATR, and you would normally go to a contract research organization and you would screen a bunch of molecules, and these molecules are the same molecules everyone else screens against every other target, just to see if there are some hits. And these hits are basically are these molecules in a library showing any kind of bioactivity against the target. And then so now you got your hits, and now you got to get rid of most of them because they're not drugs. Uh, they're reactive. They're various other things, and so you spend that time to triage, and then now you've got to start optimizing other properties in order to make it more like a lead. You've got to make it drug-like. You've got to look at physicochemical properties like solubility. You've got to look at the size of the molecule. And this is especially important when you're trying to cross the BBB and trying to make a brain-penetrant molecule, because that's all chemistry. And that's one of the areas where small molecules can really stand out versus antibodies, right? For example. So, when you look at sort of oncology as a space, where small molecules, in my opinion, can really differentiate is in, getting into the brain. So that aside, brain penetrance, the physical chemical properties, and then the selectivity against, these off targets, these things are all things that people would normally do sort of in serial. So you find your bioactive molecule, then you take care of your phys chem properties, then you look at selectivity, and then finally you want to make sure that they're novel, that someone hasn't already patented these things. Every single property requires a different type of optimization, and when you do these in serial, you often will get it's a lot harder to get to where you want to go. Whereas with us, we put all of these properties in at design time, and then boom, we do native, what we call multi-parameter optimization, which is very hard to do. And that's because generative models, when you think about generating text or generating images, that's what they do. They do native multi-parameter optimization. Your prompt defines a number of properties, and it will simultaneously take all these properties into consideration and then generate an output that meets all of the desired properties. So when we got the target product profile from Rakovina, we were able to really use that in order to generate these structures. And that's really the standout. And that's how we got to where we are today with these compounds. And most importantly, these are novel, right? These were not compounds that were sitting in a library or in a virtual screen or a make-on-demand space, regardless of however many tens of billions or even a trillion. I mean, chemical space is vast, and that's the potential space of all drug-like molecules. It's like 10 to the 60. It's absolutely massive. And get a trillion sort of molecule library, that's still 0%. A trillion times that is still 0%. So the best way is not to search through chemical space, but is to actually start from what it is that you want and then design it and using Enki. And that's really what we did. And I think the results speak for themselves.
So last January or so, we had Rakovina receive the initial output from Enki, which was really generated in the speed of light, which is the whole point of the cloud. As we started this in the fall, and to be able to go in parallel with those different parameters and deliver to Rakovina what they were looking for, and in MADS, Rakovina received that short list of compounds, and then over the next several months leading up to the presentation at the Society for Neuro-Oncology meeting, walk through that process and where we are now.
Yeah, so basically when we received the list of molecules proposed by Enki, the AI, then we put them into chemical synthesis. So we make the compounds as suggested by the AI. And when we receive the compounds, then they go into a variety of functional assays to really validate how good Enki was to predict the features that we wanted in the molecules. And those assays are, first of all, can they inhibit the target proteins? Do they have an impact on viability of the tumor cells? Are they stable enough to be drug-like? Do they have a pharmacokinetics profile that is workable in future optimization? All those kinds of things. And I have to say that Enki, the hit rate coming out of Enki was extraordinary. And I think my guess is like 30 to 40% of the molecules that we synthesized and tested, they basically hit the majority of the targets features that we wanted in the molecules, which is, I've never seen that before. It's really extraordinary.
Just to be just a little editorial, that's actually lower than what we usually get. We're above 50%, but this was a hard TPP, so we're very proud of that. Yeah.
It was because we wanted We wanted both to target ATR and mTOR, but we also did not want the molecules to target some very closely related kinases. We wanted to avoid touching them, and that, of course, made the whole project a little bit more complicated. But anyway, so that I just want to make a quick comment on why we want to target both ATR and mTOR. And that's really related to the patient population that we want to position this drug for. We want to position this drug in patients that have loss of function in a tumor suppressor called PTEN. And the reason for it is that PTEN deficient tumors, they have a high level of what we call replication cell stress. And that is the type of cell stress that creates a lot of DNA damage and DNA stress when the cells they proliferate. And that's why PTEN deficient tumors are sensitive to ATR inhibitors. So when we inhibit ATR, then we can create a synthetic lethality scenario where we can eliminate cancer cells. But another consequence of PTEN-loss is an activation of a survival pathway that converts to mTOR activity. So that creates a potential escape route for tumors treated with ATR inhibitors in this context. And that's really the rationale behind taking out both mTOR together with ATR at the same time, because then we can close that backdoor, potential backdoor that tumor cells would have escaping that treatment. So anyways, the data that we have so far has validated our approach very nicely, and it really confirms the power of Enki.
And I think it's worth highlighting that PTEN story. The tumor suppressor genes are very important in terms of maintaining our own homeostasis and preventing cancers from happening. And for those of us who've been around for a little while, we will recall that back in the early 90s, I think maybe 1993, that the journal Science sort of mirrored what Time magazine does. And their Person of the Year, and they selected a tumor suppressor called P53 as the molecule of the year. And it really led to a lot of development and excitement and focus in terms of cancer research. And PTEN is arguably as important, but it has not had the highlighting and the focus. And for two young companies to be sitting here saying, we've got something that is unique, that is frankly in a leadership position at another major tumor suppressor gene that is as important as the 1993 molecule of the year is pretty extraordinary. And to go from an idea to being able to present data initially at the AACR-NCI EORTC meeting, which is a big meeting of cancer organizations from around the world, where we really showcase for the first time the work that Variational and Rakovina have been doing, leading up to the Society for Neuro-Oncology meeting, I think is extraordinary. And the gravity of the potential impact on patients and leadership position to do it is something that we're all very proud of. And it's been an amazing path to get here, and the steps forward are going to even be more exciting. Matt, talk a little bit about that triple meeting presentation and how you think we as a collaboration stood out amongst the peers of ATR presentations, and then let's talk about the SNO meeting and where we go from here.
Yeah, so the AACR triple meeting in a few months back was the first time we really talked about the ATR program from Rakovina, at least with functional data behind it. And there was a lot of ATR interest at that meeting. I would say a few years back, the main interest was on other DNA repair proteins, but this year it was clearly ATR that had the interest of the industry. And there are three main ATR candidates in clinical development at the moment, at different stages. And they are used in combination with other treatments that are already approved. And the data look really good. The problem with those inhibitors that are in clinical development by other companies is that they are not able to penetrate into the brain and the CNS system. So the interest for an ATR inhibitor that can penetrate into the brain was absolutely extraordinary. We had clinical teams from the main clinical trial units in the world, both US and Europe, coming over and asking if they could take our new inhibitor into clinical trial when we are ready. So the interest was absolutely extraordinary. And at the SNO meeting, which was actually only like a month later, at the SNO meeting, we were able to basically solidify the strategy on how we wanted to position such an inhibitor that could target mTOR and PTEN at the same time. So at the SNO meeting, that was the first time we really put forward the argument for targeting PTEN-loss tumors, patients with PTEN-loss. And that rationale was received extremely well. And lots of collaborations were established where we will get access to interrogating tumors of various origins for PTEN-loss and biomarkers. That would be extremely helpful for us in future development of this molecule here. So all in all, I would say that ATR is a hot topic at the moment, and going the route of brain penetration seems to be something that resonates extremely well.
Yeah, for those of y'all who haven't, had the opportunity to attend these big scientific meetings, the meat of the meetings are poster sessions where you literally have 100 different or more different presentations happening simultaneously, where the researchers, in this case Mads, is presenting the results of the work. And at that meeting, the triple meeting, as we call it, in Boston a couple of months ago, there were dozens of ATR presentations. It's a hot area, but there was really only one that stood out because of the blood-brain barrier presentation. And you can always judge how much interest is by the number of people that are standing there at that poster and trying to understand the data and learn about it. And also, at the end of the discussion, because there's a period of time where that session's over and you move on. If Mads is stuck there for another 45 minutes beyond the end of the session, that's a sign of interest in the community, and that happened. So it was a very exciting to watch, a program that has gone from idea to validation, and a type of validation of the companies like big pharma companies who have ATR inhibitors in the clinic that don't cross the blood-brain barrier, where they've invested hundreds of millions of dollars to get there, to have the collaboration between our team and Variational deliver data on par with this in that short of time is pretty amazing. So we have some great candidate molecules that have come out of this, and we're going to take some next steps to confirm a lead that we'll look to move toward clinical trials. And so let's in the few minutes before we turn it over to questions, why don't we talk a little bit about that process and how we're going to make it happen.
Yeah, you want me to start there? So basically, the situation is that we now have functional data on the inhibitors or the structures that was proposed by Enki. And now we have a good handful of molecules that are at the top of the list, that have various degrees of performance in the parameters that we want this molecule, the lead molecule, to have. So what the task is now moving forward is really to see if we can, combine all these fantastic properties in the top five molecules into one clinical lead. And for that, we will continue our collaboration with Handle and Variational AI and go into a new phase, which is optimization or lead optimization. And perhaps Handle, you can explain better than me how you run that.
Yeah, so we're excited to be able to move to the next phase in lead optimization and prove Enki. And it's a somewhat related yet slightly sort of a left-hand, right-hand sort of way that we use Enki. So in the first task where we did lead generation, and these are the molecules and the leads that you have right now, the task for Enki was to trained on all of the data that it has been trained on, is to come up with these novel chemical structures that met all of the parameters that were in the target product profile. And so really the task was to generalize and extrapolate into different areas of chemical space that you wouldn't be able to get through, efficiently via screen or using sort of scaffold hopping techniques. But now that we have these good leads, what we plan to do is constrain the scaffold of these leads, and Enki also can do very, very efficient local chemical search and do it in a very efficient manner, very similar to what a medicinal chemist would do. But it's really a tool at the hands of a medicinal chemist to vastly accelerate the ability to optimize these leads. And it's very hard for a human to keep, as skilled as they are, to keep everything sort of in their minds. And the way we like to say is, imagine Enki is a very dull student that doesn't know anything to begin with, but has perfect recall. And once you've trained Enki on basically everything, and it has perfect recall, then it suddenly seems to have these sort of like ungodly powers. Well, it just remembers every single thing. Whereas a human, regardless of how skilled and how smart you are, you're going to forget things. It's impossible to hold all of these things in your head while you're doing sort of the optimization of the leads. And really in Lead Optimizer, what we're doing is we're saying, okay, now hold this scaffold or scaffolds, and then boom, do that efficient chemical search and get you to that, optimized lead so you can nominate that drug candidate far more efficiently. And we've done this before for other customers, and we're doing it right now for others, and we're raring to go to apply Enki on the lead optimization with Rakovina.
Let's talk a little bit about the expected sort of timelines and output, because we're all used to having drug development and this optimization process take months to years. And what we're hoping to achieve collaborating together to get to that lead molecule, we're targeting obviously much shorter than that. But talk a little bit about steps and time and process, and what we should all expect to be sharing with the world if we were here again 6 months from now and a year from now.
I mean, you want to get to a DC, a drug candidate, right? And so you need to hit a certain number of parameters, and what defines an optimized lead is what Rakovina has defined. The great thing though is that because we started with Enki Lead Generator, we're a lot closer to an optimized lead than you would be given the same amount of time and effort. Now, for everyone on the call who's experienced in drug discovery, that lead optimization is the longest and the hardest phase of drug discovery. Very often, lead optimization programs stop when you run out of budget or you run out of time, and you keep going until you can't go any further, or you're successful. Sometimes you get lucky and it can be done in 6 months. It's extraordinarily rare. Sometimes you spend 3 or 4 years, and it doesn't matter if you're a biotech or pharma, and you never get there, and you just like have to pull the plug because you're just burning a lot of money. So really for us, what we're doing is we're already closer to sort of the finish line on the DC. So for us to do the lead optimization is a lot easier to do. The distance between where we are now versus an optimized lead is a lot closer than you would normally find. I think given the way that the current leads perform versus these clinical stage assets, these competing programs from pharma, I think we're starting off. Our starting line is not back here. Our starting line is over here. So you have that, and then in addition to that, you have the vastly efficient way that we've worked on the program. The awesome thing is we've already built the model, and so the model's already generated these leads. And so for us to now take that model in Enki and find even better and more optimized compounds is relatively straightforward. So I don't want to commit to any specific timelines, but I guarantee you this: it's going to be a lot faster than if you didn't use it. And we're gonna. The other thing is oftentimes you find that a lead series will stop in lead optimization. You get to a stage where you've optimized it as far as you can go, and now you've painted yourself in a corner. That lead is no longer developable, right? You've reached what we call a local minimum. So, and you're trapped there, and it's very hard to get out of it. And then you kind of have to go back to your backups. The good thing is we've got like several really good backup series as well. So in the event it doesn't work out, we've got these other scaffolds that we've generated as well. So we can start working on multiple scaffolds at the same time as well, which not only decreases the time and is more efficient, but also decreases the risk. And that's really the true power is at Variational, we know what we're good at. We're good at discovery, right? We're not good at taking a drug and then, getting into humans and doing the trials. That's what Rakovina is good at, and that's what you guys do. But we're happy to be able to deliver these compounds to you in record time, and also differentiated novel compounds. And that's really what Enki does.
And from a biology lab perspective, what really takes the longest time is the synthesis of the molecules. It can take anything from 6 weeks to 3 months, and some molecules we have to give up on because it's simply not possible to synthesize them. So that's really the time-wise uncertainty in our pipeline, because all the assays to qualify a lead we have up and running, and the turnaround time is very fast. In vitro assays is done in like 4 weeks, and then of course we have to qualify in animals, which will take another couple of months, 2 or 3 months. But it is really the uncertainty about the timing of the chemistry that we have to deal with.
Well, I think that the one thing that I know when we were all sort of talking about the optimization program recently, one of the things that's different here in this process versus the initial work where, Enki delivered a set of compounds and they were all synthesized, and then we tested them. In this case, we're going to integrate Enki with the chemistry straight away, and it'll be smaller bites at the apple. It won't be hundreds of compounds. It'll be a short list because of where we are closer to that goal line. And so the chemists will get the output of Enki directly. And so that will help to speed up the timeline. It will also be able to give feedback, not only from the biology side as Mads and the team are validating and testing in the lab, but also from a chemistry point of view, because, if it is something that's impossible to synthesize, let's know right away and stop trying to do it, So, I think we've got a great opportunity here to do something wonderful for patients. ATR is obviously a very hot area. I believe, we all believe where we are in terms of brain penetration and the dual functionality with mTOR in that PTEN space that is so important for so many tumors is a wonderful place to be. And we're looking forward to taking the next steps. So with that,
just to double click on that, Jeff. Proof is in the pudding. I mean, we just delivered, I think, 100 plus molecules to you in the lead generation phase. Contrast that to 2,000, 3,000 molecules that are generated. I can guarantee you pharma is probably closer to 3,000 than they are. But to get to where we are with 100 plus lead compounds, and I think only a fraction of those were synthesized. So we're talking about tens of compounds, and to get to this stage, that's really remarkable, and that speaks to the massive efficiency that Enki brings. And then to your point as well, and when we do the lead optimization, it's going to be like less than 10 each cycle, with each scaffold that we go through, and we'll do multiple sort of iterations, but each iteration gets better and better and better. And so at the end of the day, there's no way we're going to be more than a few hundred compounds synthesized, and that translates directly to the bottom line on FTE costs and CRO costs. Of course, CROs are happy because you're at a drug candidate, and that's really what they're there about. And so we think that this is going to be a really, really amazing use case and proof point for both of what we're doing, and we look forward to ATR as well as others.
Absolutely. So I want to open it up for questions in the time that we have left. There's one that's come across in the chat. I think we've touched on this, but Mads, talk a little bit about how the lead molecules and the target product profile that Rakovina and Variational have been working on. How does that compare to competitive molecules like ceralasertib and others that are in the clinic?
Yeah, so there are different ways we can compare to competition. The first and perhaps most obvious way is to see how well they inhibit the enzymatic activity of ATR and mTOR. And if we look at that, we are definitely equal or better than all of the three major molecules that are currently in clinical development. And then there's, of course, the dual inhibition of mTOR together with ATR, which is kind of a unique feature that we are exploring. And that is another differentiator that is important for us. And lastly, there's, of course, the brain penetrance, because the current competitor molecules for ATR, they do not penetrate the brain very well. So that's really where we think we can make a difference.
There's been another question coming from Maya. Thank you for this. Great question. So I'm not sure if y'all can see this, but I'll read it so everybody can see it. Given the focus on PTEN-loss driven brain tumors and dual ATR-mTOR inhibition, how does the AI platform evaluate the risk of adaptive resistance through alternative DDR or cell cycle pathways such as ATM, DNA, PK, WEE1, et cetera? That would help to ensure that we're going to have durable responses, because obviously the DDR system is very complex.
Yes. So that's a good question. And of course, how well a dual ATR mTOR inhibitor will perform as a single agent in a human is still, something that we simply don't know. What I can say is that the ATR inhibitors so far has been mostly combined with other inhibitors or radiotherapy. And ATM is for sure one pathway that can at least partially compensate for some of the activity that ATR is exerting in the DNA repair system. DNA PK also has some compensation. What I think is the strongest evidence that it might not be such a big issue is the fact that it was a study that was published in 2001 where they were screening for genes that was somehow involved in creating resistance to ATR inhibitors. It was a study by Stephen Jackson's group. And what they saw there, so they were looking at ATR inhibitors and sensitivity and resistance in the context of ATM status. And PTEN specifically here was picked up as a gene that highly sensitized to ATR inhibition independent of ATM status. So that provides me with some confidence that a compensation pathway with ATM might be manageable. But of course, we won't know until we have it in humans.
Makes sense. If there are any other questions, please raise your hand or type them into the chat. Otherwise, we're really appreciative. Couple follow-up questions just came in, so perfect. Can we talk about the business strategy here? How long to optimize the candidates, and sort of what we would hope, where we would hope to be, for example, at the AACR meeting in the spring, which is really the next major cancer meeting that I think Rakovina will look to present. So, Matt, do you want to talk a little bit about that?
Yeah. Let me just understand the question here correctly. Yeah. So basically what we do is that we work together with what we see as the best and most competent chemical vendors that we engage to actually synthesize the compounds. And we also in Rakovina have medicinal chemists, and Variational AI also have medicinal chemists that are always standing by to help making sure we are able to synthesize the right and correct chemical compounds and that they are validated correctly. And of course, the brain penetrance and permeability is something that we always will have to test functionally in the lab. And we do that we have in vitro assays that can do that, but we also usually do the final testings in animals to get a real-time readout of brain penetrance. In terms of what was the last part of the question here? Novelty BMA in both hit to identification and hit to lead stages. Yeah. So
I can answer that.
Yeah, if you can answer that, Handel.
Yeah. Okay, fun one. So again, all of these properties that you mentioned, synthetic accessibility, novelty, BBB penetration, permeability, efflux, MDC, like all of these properties, whether they be phys chem properties or whether they be, selectivity properties in terms of defining on targets and off targets. And going back to Mia's question, we've trained on ATM, DNA, PK, and we won, right? So it's just how you tune that TPP. If, for example, it's required that now we need to be, we need to hit a third, on target versus, being selective against all of these off targets, that's really quite simple for us to do. And we've done that before. But we focus on the biological thesis that Matt's already outlined. But, in the event that we need to come back and revise that, we're the fastest way to do it. We've already trained on it. So it's just basically another Enki run. Going back to Fan Wang's question, all of these properties, in these and more, including PK and tox, these are done simultaneously. So there's no sort of trade-off in the sense that you would normally do when you're doing a serial hit identification, or hit to lead rather, or lead optimization. Everything is considered in tandem. And the reason why we do that is getting into the sort of the machine learning bit is these are high dimensional problems. Each property relates to a specific type, it's a dimension. And when you're looking at various, the energy landscape of chemical space in terms of, these various properties, what we're essentially doing is we're taking this very high dimensional problem, which is multimodal, and then we're basically projecting it into what's called a latent space. And then within the latent space, we transform that multimodal representation to a unimodal representation, where we're able to do much more efficient, gradient descent, such that we are able to get as close to a global minimum across multiple properties all at the same time. Then we unwrap out of latent space back into chemical space, and we end up with different structures. That's one sort of crude definition as to how we do it. But all of these properties and more, there are about 30 or so properties that we optimize for. These are done simultaneously, and that's the power of the machine learning, and that's the power of Enki. Sorry.
Yeah, and if there's like one aspect that we would like to have in the molecules that underperforms a little bit, Enki will know it right away because it's an iterative process, and we will feed the information back, and so
But again,
that
yeah, I mean, you have to tell Enki what you want, right? So
yeah.
it's not like it's not an agent that kind of like says, oh, look out for that. I mean, it's very much a tool used by chemists and in the service of biology. So we're not there yet.
But I think where we are is, in having this set of leads that we presented on at the Society for Neuro-Oncology meeting is, it's an amazing place to be from where we started less than a year ago. And to have, that level of enthusiasm from the scientific community, and knowing how closely we believe we are to taking where we are and making it into the molecule that we want to partner, that we want to take into the clinic, and having a straight line shot through Enki to get there in the most efficient way possible is I think everybody can tell the enthusiasm of the three of us, and I can say that's echoed to the rest of the teams that are working on this together. Mia, it looks like you have your hand up if you have another question.
I do have another question. I wanted to turn on my camera, but it's blocked. That's okay. Thank you so much. No, it's okay. All good. Thank you so much for answering my first question. The second one I have is, do you see your approach ultimately being used as a monotherapy or primarily in rational combinations with radiation actually, or chemotherapy in brain cancer?
Yeah, so thank you for the question. I think the positioning in a PTEN deficient background really creates an opportunity for monotherapy. But what I can tell you is that the clinical trial units that we are in contact with in North America and in Europe, many of them, their interest lies in combination with other agents. So, and where we're going to start is really, I cannot really predict that yet, what will be the starting point. But I think there's a very strong chance that this could be a monotherapy, and for sure it could work in combination with other chemotherapies and radiation therapy. One interest that we had was combinations with telazolomide. That is basically the only chemotherapy that you have for brain cancer at the moment.
Yeah, thank you.
And of course, the PTEN story goes much beyond brain cancer. And, the way that the opportunity here to work with leading academic medical centers who have frankly approached us, approached Rakovina, approached this collaboration based on what we have so far to say, look, we would like to be front and center as this moves into human clinical trials. And there are opportunities to work together in preclinical models to really refine what those combinations might be. So stay tuned for some, I think, additional scientific presentations at major meetings through collaborations they're going to develop in the early part of 2026. So if there are no other questions, thanks everybody in wrapping up. Mads, Handel, thank you for taking the time to share the story of the collaboration and the success that's happened to date. And we're all very enthusiastic about the next steps and look forward to sharing that with this audience and the future. And thanks everybody for your time. And please feel free to reach out anytime with any further questions. Have a wonderful day.